Good evening, Professor. Good
Yes.
evening, Professor Kruger and Professor Purushottam. Good
evening.
evening.
Yes.
Hello,
Yes.
hello.
Hello.
Nice meeting you.
Yeah,
So
same. Great.
Arun belongs to the Department of Chemistry. He's on the faculty there at IIT Tirupati. But I wish he belonged to the Department of Physics as well. Because what he does is as much physics as chemistry. He does density functional theory calculations and quantum chemistry in general, statistical mechanics. So it's very much a part of our physics domain. And Arun will also help Purushottam in conducting the question answer session. Purushottam.
Yeah.
Gladly, yeah.
So it's wonderful to have Arun on this for this session.
Thank you, Professor. Yeah, it's great to be here. I mean, nowadays all the work is interdisciplinary.
Yes, absolutely, and that's very true.
And I see, Professor Kruger has, work extensively on the light harvesting complexes, the excitonic behavior. And I'm, eager to, listen. I mean, that too also from the experimental tool, you are able to catch up the single molecular level phenomena. That's quite exciting. Same.
Yes, that's true. And I completely agree that we have to focus on multidisciplinary research and forget about the distinction between chemistry and physics.
Very true.
Because at that level,
yes,
there is no distinction anymore.
Absolutely. Absolutely.
You also see that I publish very often in journals of the American Chemical Society.
Yes.
So yeah, is it chemistry or physics? Well, I would say it's both.
Yes, absolutely.
Absolutely.
Yeah, it's a highly reputed journal. Yeah.
Yes.
Yeah.
So it's about time that we start. So shall we?
Yeah, it is now exactly 2 o'clock in South Africa and 5:30 in India.
So if you're okay, Professor Kruger, we'll begin the formal session.
Yes, I'm ready. Thank you very
Excellent.
much.
Excellent. So good evening to everybody here in India, and good afternoon to Professor Tjaart Kruger. A very hearty welcome to today's seminar. We have a very distinguished speaker, and we are already well past 20% of this century. And in this century, there are two big things which are happening. Everybody talks about the second quantum revolution because quantum mechanics has certainly been used extensively in the last century to build semiconductors and quantum devices. But quantum entanglement is now being used to build quantum computing technology. So that's a big thing in this century. And the other big thing which was predicted of this century sometime back was advances in quantum biology. And just now before we started the session, we were in agreement that science is very interdisciplinary, and it is important to appreciate problems, methodologies, solutions from different fields so that we can really push the frontiers of science. And we have a very accomplished speaker today, Professor Tjaart Kruger. And I would like to invite my longtime friend and colleague, Professor Purushottam Chakrabarti, who is the condensed matter physicist, well known for extremely important experiments that he has done. But I will not spend much time on that, but I would like to invite Purushottam to please introduce our distinguished speaker today. Purushottam?
Yes, I'm ready. Yes. Am I audible? Yes,
Purushottam. Yes.
Okay. Good afternoon. I can say good evening here. This is my pleasure to invite Professor Tjaart Kruger of the University of Pretoria, South Africa. My pleasure, secondly, because I had been at this university for quite some time, and number of times, in fact, at the physics department of the University of Pretoria, as an adjunct professor and as also an honorary professor. Well, and recently I met Professor Kruger at the South African annual conference just 2 months back in one of the place of South Africa. Well, Professor Tjaart Kruger completed his master's, which is called cum laude, in computational space physics at the Northwest University in South Africa, and PhD in experimental biophysics at the Vrije University in Amsterdam, in Netherlands. He continued working on the same topic during a short postdoc fellowship at the same university, after which he returned to South Africa to establish his own research group at the University of Pretoria, where he is still based. Professor Kruger is currently an associate professor at the physics department of Pretoria University. Professor Kruger's research focuses mainly on the primary processes of synthesis, which he investigates using various laser spectroscopy techniques and some theoretical and computational. He is particularly interested in the design principles of photosynthetic light harvesting complexes and how these principles may be applied to improve crop yield, or the design and performance of organic solar cells. His another interesting area is in finding ways to control light harvesting processes using tools such as shape light and plasmons. He has authored and co-authored nearly 60 journal articles, 3 book chapters, and an advanced textbook on optical microscopy. Keeping in mind the role of science in society and societal responsibility, Professor Kruger has written several popular science articles spoken on a national radio station in South Africa. Professor Kruger's scientific contributions have received broad recognition by several organizations in South Africa. He is a member of the South African Young Academy of Science, chair of the South African Biophysics Initiative, and a senior editor of the American Chemical Society's prestigious journal, Physical Chemistry Letters. With this short introduction, I would request Professor Tjaart Kruger to start his lecture today. He will be talking on the physics of photosynthetic light harvesting. Professor Kruger, please.
Yes, thank you very much for the kind introduction and especially for the kind invitation to present at this prestigious seminar series. Yes, so I understood that the physics is that the audience is mainly a physics audience, and that was the reason why I chose this specific title for my talk, the physics of photosynthetic light harvesting. This is by definition a multidisciplinary subject, but I would like to focus today specifically on some of the physics aspects, and I will only be able to touch on some of the aspects because there's so much beautiful physics from many points of view that one could look into this subject. Okay, so let me start off by just showing you on a map where Pretoria is. Pretoria is not far from Johannesburg, which most people know. And in fact, Johannesburg is not the capital of the country, but Pretoria is, or more specifically, it's the administrative capital of the country. And September is a beautiful month of the year because we have a lot of jacaranda trees, and this is the time in which they start to blossom. So towards the end of September and October, you will see a lot of these jacaranda trees all across the city. And because of the prevalence of these trees, Pretoria also has the nickname called the Jacaranda City. Okay, here's an aerial view of some part of the campus of the University of Pretoria. I am in this building called the Natural Sciences 1 building, and here's another building just close by our building, just to get some idea about the student life and the beautiful flora that's maintained at the university. Okay, I would also like to give a quick overview of my research group's activities. So we are focusing mostly on photosynthetic light harvesting complexes, and the motivation for me to study these systems is because of all the very interesting physics that takes place in these systems, in a biological system, and then also the broad range of applications, so that one knows that your research also has potential for long-term technological applications. One of these applications is in the field of solar cells, and I'm especially interested in looking at design principles of photosynthetic light harvesting complexes and how they may serve as a source of bio-inspiration for especially organic solar cells. Then we are working mostly on experimental research. Our main experimental method is single molecule spectroscopy, but we are also doing transient absorption spectroscopy to resolve dynamics of the energetics in these systems on a femtosecond to picosecond timescale. And then we are also looking at theory and simulations, which forms a basis for interpretation of most of the experimental results. Okay, our main purpose of the research is to understand how nature works. We want to know how nature works at the most fundamental level. And in the process, obviously, one discovers new functions, new features of these systems. One way in which we investigate these systems is to develop different methods, mainly different single molecule methods. And one goal that I have is to develop methods that can enable us to do single molecule spectroscopy in a live cell. Okay, and then on the other hand, we're also using different ways to manipulate and control the different photophysical processes. Okay, so here's also a summary slide. I'll be very brief. So our focus is on fundamental research of light harvesting systems, firmly grounded in theory and simulations. And here are some primary applications that we are not looking at ourselves, but this is always, we always keep that on the horizon to know that our research also has significance in the technological sector. So some primary applications and some secondary applications here at the bottom. And I would like to highlight just one application. So based on decades of research on especially the primary processes of photosynthesis, the research community has now gained enough knowledge that genetic engineering of some aspects of the photosynthetic process can be done. And it was demonstrated on different types of crops. It was demonstrated that by genetically engineering these crops, the photosynthetic yield could be enhanced by as much as 33%. And this is really extraordinary for crops out in the field. And this is because of our knowledge of photosynthesis and especially the primary processes of photosynthesis. This is one tangible application. Another emerging field, but tangible application, is the early diagnosis and treatment of diseases, which can be done by looking at fluorescence, photoluminescence, during the primary processes of photosynthesis. And this can also save a very big percentage of crops before the symptoms of diseases show up normally after a few days. Okay, I also understood that the audience doesn't have a lot of experience with biophysics, so I thought of introducing the audience to biophysics from my point of view. And the definition I am using is a fairly simple definition. So let's consider any biological system. Okay, this is the context in which we work. We want to look at some biological question at the quantitative level. So what a physicist does, or specifically a biophysicist, is to take the lens of physics. And this is the methods and principles of physics, the toolbox of a physicist that is used in order to look at the biological system and in order to look at it at a quantitative level. So this can involve experimental work, developing models, doing simulations, everything that physics allows us to investigate these systems at a quantitative level. Okay, I would also like to offer 3 motivations for why biophysics is very interesting for me. First of all, physics brings the understanding of biology to a deeper level, and that is illustrated, for example, in this one editorial that was published 10 years ago in Physical Biology, where a plea was made to theoretical physicists to help the biologists understand their data better. And last year, I organized a conference where Dr. Martin Friede from the World Health Organization was one of the speakers. And one thing he said caught my attention very vividly. He said that you will not be able to successfully develop vaccines without biophysics. And this is especially after coming out of the COVID-19 that has wreaked havoc across the world, and everyone knows the importance of vaccines. So in order to develop the next generation of vaccines, biophysics will play a crucial role. Okay, even nature physics has an eye on biophysics. As you know, nature physics focuses mostly on physics, but there's a lot that can be understood by studying biological systems. And in this article, it was said that a lot of biologists make a straight appeal to physicists to help them make sense of their data. So just highlighting again the importance of multidisciplinary work, because it's especially when we are crossing disciplines or when we are putting different disciplines together that a lot of our critical questions and cutting edge research questions are answered. Okay, second motivation is that biology offers a test bed for some theories of physics. One example of that is non-equilibrium statistical physics, for which biological systems, small biological systems especially, are an excellent test bed. So Christopher Jarzynski, who wrote this commentary, he's known for the development of fluctuation theorems in quantum physics. And especially small systems, quantum systems are known to be excellent systems to investigate these fluctuation theorems. Last year, I published a paper together with a colleague in Physics Reports where one aspect that we looked at was these fluctuation theorems. And we looked at different thermodynamic processes ranging from different quantum systems, atomic clusters, all the way up to stellar galaxies and cosmological models, where we looked at thermodynamic processes and also investigating, for example, the role of fluctuation theorems and so on. Okay, the third motivation I would like to offer is that biophysics drives critical innovative developments. And this term, century of biology, was coined in this context of innovations. So you're welcome to read up more about that, but it was stated that this century is the century of biology. A lot of biological developments in science leading to innovative developments in technology are predicted to happen in this century. And when it comes to innovations, technology, that's where physicists again play a crucial role. Okay, then there are many different areas in which biophysics contributes directly or indirectly to. So here's a list of them. Earlier this year, I've written a status report on the African biophysics landscape where we have spoken a little bit on some of these applications and especially how we envisage the development of biophysics on the African continent in order to address many of these global issues that are especially important for a poor continent like Africa. Okay, quantum biology, which was mentioned during the introduction, that is an emerging field, a very interesting field. And I've shown here a table of key enabling technologies that the EU is trying to develop, different research fields in which these processes on which these processes are based, and examples in nature where systems or organisms in nature can do what we would like to achieve in our technologies. In other words, there's also a lot of bio-inspiration, even biomimicry, by looking at natural systems at the quantum level. So how systems use non-trivial quantum effects or just achieve certain results by using non-trivial quantum effects. So there's a lot that we can learn from nature. And I've here encircled light harvesting complexes, photosynthetic photosystems, to show you that in photosynthesis, we can already achieve at least 2 of these technologies by understanding these systems at a deep quantitative level. Okay, I would like to say a few more words about quantum biology. Here's a headline review that I co-authored together with others a couple of years ago, and I would heartily recommend this, where we have looked briefly at the history of quantum biology, but especially we wrote about the current state of the art and what we predict will be the future, the near future of quantum biology. So Philip Ball is a scientific journalist, and he wrote a newsflash article some years ago in which he said that the key to practical quantum computing and high-efficiency solar cells may lie in the messy green world outside the physics lab. And here he especially had in mind photosynthetic systems. So photosynthetic systems, one would not expect quantum effects to play any role there because they are, as he describes them, a messy system. So at room temperature, there's a lot of noise in the system. But some time ago, it was proposed that in these systems, there are long-living quantum coherences that are sustained for times as long as a few picoseconds. So that is why people became interested in looking at the design of these systems and how that might be applicable to the field of quantum computing, where you can sustain coherences for very long times. High-efficiency solar cells, this application is based on the fact that photosynthetic systems, especially the light harvesting systems, are extremely efficient. So the light of every absorbed photon is used under ideal conditions. So there's a lot of design principles that we may apply to our solar technologies, for example. Okay, here are a few systems that have been proposed to use non-trivial quantum effects in order to establish a biological function or significantly enhance a biological function. And the most vivid and I would say proven examples of quantum biological systems, so the use of non-trivial quantum effects, is in the field of photosynthesis. So this is again one reason why I'm very interested in these systems. Another system is magnetoreception, which I would also say has been proven to a very good extent, that some organisms, insects, birds, use non-trivial quantum effects in order to give them a sense of direction. And I wanted to illustrate this very briefly. So let's consider, for example, this European robin, which flies very long distances, from close to the North Pole to the Southern Hemisphere and back again every year. And in the back of the retina of this bird, so on the retina, at the back of the eye, there are a few cryptochrome molecules, and there are actually 4 different types. One of them, there has been significant evidence that one of these cryptochromes is involved, was very highly likely to be involved with magnetosensing. So the ability of this bird to know which orientation to fly when it's making these very long haul flights. And how this mechanism works, it was actually proposed by Klaus Schulten already in the 70s, long ago, but obviously the experimental work lagged behind very much just because of the sheer difficulty to experimentally test these ideas. So how the model works is that there's a donor and acceptor in the cryptochrome, and upon photon absorption, a radical pair is created. So we can divide this process into 3 steps. So step 1, there's a photo absorption, and then we have a photoexcited neutral donor acceptor molecule. But immediately after absorption, an electron will be transferred from the donor to the acceptor molecule, but these 2 molecules will still be coupled. So they form a radical singlet state between this coupled donor acceptor molecule. Now, this singlet state can interconvert to a triplet state and back due to both an internal magnetic field, or a hyperfine interaction, and also due to external magnetic fields, interaction with the geomagnetic field. And this interconversion between these 2 states, there's significant evidence that this is sensitive to the frequency of the magnetic field, it is light dependent, and it also gives the bird orientational dependent. So I think there is significant evidence that this non-trivial quantum effect is at the basis of magnetic field sensing broadly across different organisms. Okay, let me now get to the photosynthetic context of the systems that we are mostly working on. And just to set the biological context, we are looking at some of these systems, most of them which are embedded in a membrane. Okay, and when we look at them from the top, we can take out, for example, photosystem 2 supercomplex. It contains a so-called reaction center in the middle, then some core antenna complexes and peripheral antenna complexes on the sides. And the main purpose of these supercomplexes or the photosystems is to capture solar photons and transport the excitation energy as fast as possible to the reaction center, where a charge separated state will happen. So similarly to what we've said here, so there will be a donor and acceptor, which in this case chlorophyll molecules, so that an electron can be transferred from one molecule to the next, and then there's a hopping process of the electron until that you have an electron that's on one side of the membrane separated from protons on the other side. So this charge gradient, actually a double charge gradient, electron charge gradient and a proton charge gradient, this is now the potential energy in which the energy has temporarily been stored. And after that, the energy will be stored in continually more stable forms of energy until the sugars are formed in the end. From a physics point of view, the most interesting processes are taking place at the beginning. So during light absorption and migration of the excitation energy to the reaction center, because there are a lot of quantum mechanical processes taking place there. So these are the systems that I'm going to focus on mostly during this talk. Okay, so let's zoom into one of these systems. So one of these peripheral systems is called LHC2. This is the main light harvesting complex of photosystem 2, and in fact, it is the most abundant membrane protein on Earth. So it's a very important system to look at. So this, we call this a pigment protein complex. It contains 3 proteins and 54 pigments. These pigments are mainly chlorophylls, but also some carotenoids. And what I would like to highlight is the very high density of pigments packed into this protein which serves as a scaffold, actually more than a scaffold. I will get back to that later. So just consider the dimensions of this system. We can approximate it by a disk with a diameter of 7 nanometers and a thickness of 4 nanometers. Now we are considering 54 pigments. Okay, the chlorophylls. Let's look at the chlorophyll. Okay, we have a conjugated ring, so double single alternative bonds between the carbons here, okay, which forms a ring, a conjugated ring system. So quantum mechanically, we can approximate a chlorophyll very well by a particle on a ring. Okay, the diameter of this ring is 1 nanometer. Okay, so it is mostly a two-dimensional system. Okay, but diameter of 1 nanometer. So here we are considering a disk with a thickness of 4 nanometers and diameter of 7 nanometers. Now imagine packing 54 of these pigments into such a small volume. Okay, this is especially what is giving rise to the very interesting quantum mechanical effects, which I would like to highlight. So due to this very high density, so-called molecular excitons are formed. And I just want to say at this point that these excitons, molecular excitons, are different from Vannier-Mott excitons in semiconductors. So Vannier-Mott excitons occur where the electron and the hole that stays behind are not coupled. Sorry, they are coupled, but they are not on the same molecule, on the same atoms. And the delocalization length is normally much greater. For molecular excitons, the electron and the hole that stays behind are always on the same molecule. So when an exciton is transported amongst different molecules, the electron and the hole will basically be transported together. Okay, so these molecular excitons are delocalized electronic excitations. They occur when two dipoles strongly interact. So if we consider the interaction between them to be much greater than the coupling between the electronic states with phonons, so electron-phonon coupling, that leads to the formation of molecular excitons. So firstly, we get an energy splitting. Okay, and secondly, we get delocalization of the electronic excitation across both molecules, which is illustrated here. So here we have the wave functions of the individual molecules, molecule 1 and molecule 2. Here, molecule 1 is in the excited state, molecule 2 is in the ground state, and here vice versa. So this is just a normal way, quantum mechanically, to denote a delocalization over both states. Okay, so this formation of molecular excitons leads to a lot of physiological benefits. And here I've listed 5 of them. So firstly, you get enhanced light absorption, and that is mainly because of this energy splitting, which is then increasing the absorption cross-section of the system. That is beautifully illustrated by, for example, this system, which is light harvesting complex 2 from purple bacteria. And shown here are only the pigments, the chromophores, so bacterial chlorophylls, which are arranged in 2 beautifully concentric circles. Okay. Now, because of the excitonic coupling, you get an energy splitting. So you get 2 bands that arise, and the distance between the upper and the lower excitons is 50 nanometers. So they are 50 nanometers apart due to this exciton splitting that is happening. Okay, so this gives rise to significant broadening of the absorption cross-section, leading to enhanced light absorption. Okay, there's also enhanced energy funneling because of the formation of these excitons. So now one gets an exciton manifold, and because of the coupling between these excitons and phonons, excitations will be concentrated mostly into the lowest energy states. Okay, and this is different from a classical system. So classically, one would use an equipartition where the likelihood of population of each energy level will be equal. It's different for a quantum system. Thirdly, there's enhanced energy transfer. So more than 10 times, and this can be modeled quite easily. Okay, fourthly, there's less trapping and energy loss of the energy in the antennas. And there's also faster charge separation in reaction centers. And one of the main reasons is because of the formation of the delocalized electronic excitations. And a way in which I conceptually illustrate it often is by comparing this with playing soccer with a golf ball. So what is the main reason why it's difficult to play soccer with a golf ball. The reason is that the order of magnitude of the golf ball is the same as that of the grass field. Okay, so if we consider all the bumps in the grass field, so the landscape, the noise in the landscape is of the same magnitude as that of the golf ball. So if you're going to kick the golf ball, it won't travel very far. It will get stuck very, very quickly. But now if you replace the golf ball with a soccer ball, now the soccer ball is much bigger than the noise in the landscape, so it will easily roll over the noise. Okay, it's the same principle with these delocalized excitations, which are now physically bigger, so they will not as easily be trapped in energy traps compared to excitations that are localized on individual atoms or molecules. Okay, so this was just to explain the concept, but now molecular excitons can form between a large range of molecules and even molecules or dipoles having different energies. Okay, and then the wave function of the molecular exciton is then simply a linear superposition of those of the wave functions of the individual molecules. Okay, and the typical delocalization length in photosynthetic systems is 3 to 4 pigments. That is because this creates a balance between the formation of the molecular excitons and the electron-phonon coupling. So electron-phonon coupling tends to localize the excitations, whereas these excitons tend to delocalize them. Okay, at this point, I would like to distinguish between these excitons, which are actually coherent excitations, and another type of coherent fluctuations that have caused a lot of hype in this research field. And that is the formation of electronic coherences, which are coherences between different excitons. So if you would excite 2 or more excitons simultaneously, if you have a phase relationship between them, an electronic coherence will form. And then in the frequency domain, you will see a beating frequency. So the amplitude will oscillate at a frequency equal to the frequency difference between these 2 excitons that are simultaneously or that are in phase. Okay, so in 2007, there was a report in Nature of evidence for wave-like energy transfer through quantum coherence in photosynthetic systems. So this is what was observed: a beating behavior in the frequency domain as a function of time, and the coherence time of this beating behavior was about 660 femtoseconds. Okay, this experiment was done at 77 Kelvin. In the following years, similar experiments were done on similar and different light harvesting complexes, low temperatures, room temperature, and even longer coherence times were observed in the order of a few picoseconds. Okay, so this was extremely interesting. The community interpreted this as the formation of electronic coherences that persist for picoseconds at room temperature in a noisy environment. Okay, and that has stirred interest in the application of this for quantum computing and some other areas. So very interesting. Okay, so this electronic coherence must be separated from 2 other types of coherences. So one gets electronic coherence between different exciton states, but there are also vibrational coherences which can give very similar experimental results. Okay, so vibrational coherences happen when there's a coherence or coherent superposition between different vibrational substates within an electronic excited state. Okay, and then there can also be a mixture between the electronic and vibrational coherences, and that gives rise to a mixed electronic vibrational coherence, which is often called a vibronic coherence. Okay, so the main question that had to be answered was what type of coherence was observed in this and a dozen other experiments. Okay, so these experiments were done later, well, actually a few years ago, much more carefully. A lot of modeling was added, and then it was concluded that these coherences are not electronic coherences. They are actually Raman ground state vibrational coherences, which means it's nothing to get excited about. Okay, but in the following year, another study was published, actually also revisiting the original system. It was this FMO complex, the same as what was done in this PNAS study. Also, the original study was revisited, FMO complex, both experimentally and computationally. And in this paper, the experiment was done a bit differently. So different polarizations were used, and then combined with modeling, they came to the conclusion that, again, most of these coherences are ground state vibrational coherences, but there might also be evidence of vibronically coupled excited state. In other words, vibronic coherences. Okay, so this means that there might also be the involvement of electronic coherences that are coupled with vibrational coherences. Okay, so the debate is not over. It's still a very interesting field of research, and yeah, probably still interesting results that will come out of this. Okay, so these types of coherences are different from the excitons that I've introduced just now. Okay, another process I would like to introduce is that of the formation of charge transfer states. And that forms when two molecules get very close together so that there's a strong overlap in their wave functions, in the electronic orbitals, okay, and such that a strong charge gradient occurs. Okay, so this can also be written as a linear combination between the two original wave functions, but now in this case, C2 will obviously be much greater than C1. Okay, so the formation of charge transfer states happens in chlorophylls when we are working in the order of 1 nanometer. Okay, so earlier this year, charge transfer states has been identified as a mechanism for chlorophyll fluorescence concentration quenching. Okay, so what is this? If we consider again this extremely high density of pigments inside, or most photostatic light harvesting systems, okay, if you would remove the protein, so solubilize the same pigments, I say chlorophylls, if you would just take chlorophylls and put them in solution, an organic solvent, at the same concentration as what they occur in the natural environment, you will get zero fluorescence. In other words, in the natural environment, okay, you get optimal fluorescence. So the quantum yield is basically 100%. But if you remove the protein and use the same concentration, the quantum yield drops to 0%. And this has been, this has boggled the community for decades. Okay, until this paper appeared earlier this year. Okay, it has long been proposed that charge transfer states were involved. In fact, in one of my review papers, I've also elaborated on this. But finally, a computational study was done, giving good evidence that charge transfer states are really at the basis for this so-called concentration quenching of chlorophylls in a solution. Okay, so charge transfer states are very important. They give rise to improved light harvesting and or photoprotection, a lot of different functions that I'm not going to go into today. One aspect that I would like to highlight is it's very interesting when these charge transfer states, so take for example this charge transfer state, when that is energetically close to the energy of an exciton state, then mixing between these two states can occur. And we can write it quantum mechanically like this, where the perturbation Hamiltonian now describes the mixing between the exciton and the charge transfer state. And then you also get energy splitting. So the energy levels are now split even further apart. And this also leads to a lot of interesting effects. So I'm going to highlight some of the effects in this talk. So the main technique that we are using is single molecule spectroscopy. The principle is very simple. The goal is to do spectroscopy on one molecule at a time, or in the case of a light harvesting complex, so this is the main light harvesting complex of plants, LHC2, the goal is to do spectroscopy on a single LHC2 complex at a time. So the light will be focused on one complex at a time, and then the excitation light will be separated from the fluorescence, and only the fluorescence will be measured. And fluorescence is then a very sensitive tool to get information about the dynamics and the photophysics that is taking place in these systems. Okay, so always when one does single molecule spectroscopy, one has to start with an ensemble average spectrum or bulk spectrum, and then compare the single molecule average spectrum with the bulk spectrum in order to make sure that the single molecule environment does not induce unphysiological effects, or in other words, drive the system into a part of the free energy landscape that is much more improbable than what is occurring naturally. Okay, so we did that for LHC2. There was a very good correlation between these two. That means that we could start to analyze the single molecule results. Okay, so the ensemble or equilibrium spectrum peaks at around 680 nanometers. Now, if you look at these complexes one at a time, okay, they switch between various different spectral states. So most of the time they would reside in a state characterized by this blue spectrum, so the blue spectrum in all these three examples, and occasionally they will then switch to a state with enhanced red absorption. So they will exhibit this red band, and sometimes there can be different red bands that can be switched on and off at different times. Okay, so we wanted to find out what is the origin of this significantly redshifted emission. Okay, and we firstly did some modeling. So to investigate the role of static disorder. So static disorder is related to the fluctuations, the natural fluctuations of the protein backbone. And now because the pigment molecules are strongly coupled to the protein, any fluctuations in the protein will be translated to fluctuations between the pigment positions, and that will lead to changes in the coupling and the orientations between these pigments. So that will give rise to spectroscopic changes. That is predicted. So here's a spectral series that is predicted by static disorder giving rise to spectral diffusion. Okay, so this is what we did. We did some modeling, and if you take the crystal structure here and here, we've numbered all the pigments, and we can get the participation ratio of the excitation in the lowest exciton state. Okay, so we can relate the spectral shape with the participation ratio of different pigments contributing to the emission. And in most cases, when you have redshifted emission, there's localization of the energy on one or more of the pigments, as opposed to the equilibrium position at 680 nanometers, where you have a much more delocalized state. Okay, so static disorder can explain spectral diffusion within about 10 nanometers. Okay, and in the meantime, we've also taken this model further because this is a complex system. It is not trivial to model them, especially if you consider the fact that the system bath coupling is pretty complex. There are a lot of distinct vibrations in the system, a lot of different dipoles that are interacting. So it is not trivial to calculate these spectra. One has to make a lot of approximations along the way. And last year, we published a paper where we compared different approximate methods to calculate linear absorption spectra, and we compared the cost and efficiency of these different methods, and we illustrated that a model, for example, what was used here, modified Redfield, is actually not a very good model to use. There are better models to use. Currently, we are applying the same principle to compare different approximate methods to calculate fluorescence spectra. And how this works, very briefly, is that one would like to calculate the linear spectrum as a function of frequency. Okay, and this is directly proportional, firstly, to the dipole correlation factor, which is the factor that is not trivial to calculate. because this includes the response of your molecular system to the incident electric field of your electromagnetic wave. Okay, and it also takes into account the electron-phonon coupling, dipole-dipole coupling, so all the interactions that are involved. One takes typically a Fourier transform of this, okay, to get all the main frequencies that are involved in this dipole-dipole coupling factor. Okay, and this is the dipole factor matrix that specifies the type of spectrum that one would like to calculate. Okay, and from this one can calculate all the different types of spectra and compare the performance of different methods. Okay, so this is what was applied in this case in order to identify or in order to explain spectral diffusion within 10 nanometers by means of energetic disorder, site energy disorder. But what about a shift like this, which is much larger than 10 nanometers? This prompted us to look at a different photosynthetic system. Okay, so what you see here is the solar spectrum as perceived on the surface of the Earth, and the absorption spectrum of LHC2, the system that I've showed you just now. Now, let's overlay the absorption spectrum of PS1, photosystem 1. So, and compare this with the solar spectrum that's transmitted through a leaf. So if you look at the solar spectrum beneath a leaf, this is what you will see. A little bit of green light that is transmitted. So the blue and the red light is completely absorbed by just a single leaf. And then the near-infrared light that is still transmitted. So now this part of photosystem 1 absorption of the absorption spectrum contributes to 40% of shade light absorption. So this has a very important physiological role. Okay, so the origin of this redshifted emission is actually connected to the redshifted emission that I've showed you just now for photosystem 2. Okay, so let me make the connection between these two systems. So this is a model of photosystem 1 antenna. Okay, then we took one component of it, actually two different components that are dimerized, so dimer of them. Okay, and then started again by comparing the bulk spectrum with the single molecule average spectrum. And here you see the extremely broad redshifted band compared to the band at around 680 nanometers that was the case for LHC2. Okay, so this is now the equilibrium spectrum that we work with. And this redshift originates from very strong coupling between two chlorophylls, specifically chlorophyll A and chlorophyll B. And this is now a case where the charge transfer state's energy is close to the energy of lowest exciton state, and exciton charge transfer mixing occurs. Okay, and because of this mixing, one gets a strong Stokes shift. That's because of large reorganization energy, and that leads to the large redshifted emission that is observed there. Okay, so what do we see when we put one such system under the single molecule microscope? Okay, this is what we observed. So most of the time the system would reside in this redshifted state, but occasionally it would switch off the red emission completely. So temporarily, a photosystem 1 system would behave like a photosystem 2 system. And what I've showed you a few slides ago is that temporarily, occasionally, a photosystem 2 system can behave like a photosystem 1 system. Okay, very interesting. Okay, so here's what one can see. So red state switched off, and then only the blue emission is still present, and this is a completely reversible process. Okay, so this switch between or actually a switching off of the red state, switching off and on, that happens due to a change in the distance between these two chlorophyll rings of 0.7 to 0.8 angstrom. Okay, which means that this charge transfer exciton mixed state is extremely sensitive to distances and to the local environment. Just a very small change in the environment can switch this red state off completely. Okay, so Frustum 1 can behave like Frustum 2 and other way around. So one can propose a generic protein structure, okay, where the equilibrium is slightly different between Frustum 1, in this case, where the equilibrium is shifted just slightly differently to put these two chlorophyll rings a bit closer together, whereas in the case of Frustum 2, the equilibrium is such that these two chlorophyll rings are on average a bit further apart. But both of them can switch to the other state by means of controlled disorder, and now the biological function is established by controlling this disorder. Okay, next I would like to show an example from diatoms. These are also very interesting organisms. Okay, so they are unicellular organisms that one finds in aqueous environments. So basically all types of aqueous environments, marine environments in the sea and also fresh water. They are very abundant systems. Their sizes scale between about 1 micron and 100 microns. So just too small for the naked human eye to see. And what you see here are different shapes and forms. So these colors are not real, just for illustrative purposes. But if you just look at all these shapes and forms, it's, this is the outer shell that is made of silica. So it is amazing what genes can make with silica, all these different shapes in these natural systems. Okay, so these are also photosynthetic systems. We extracted the light harvesting complexes. And by doing that, doing single molecule spectroscopy on these systems, we discovered a new light harvesting state in these diatom light harvesting complexes. Okay, and that is because of reduced excitonic coupling enhancing light harvesting. Okay, this is a counterintuitive statement, because what I said earlier on is that excitonic coupling enhances light harvesting. So one would expect that when enhancing the excitonic coupling, that would also enhance the light harvesting function of these systems. But now we've identified a new light harvesting state. So the reduced excitonic coupling makes provision for new light harvesting state, actually 2 new states to form, which we have identified. And we have also modeled this system. And the only way that you can explain these new spectroscopic states is when there is reduced chlorophyll A coupling. So these 3 chlorophylls are part of the terminal emitter domain, and they give rise to basically all the fluorescence. Okay, and when one heavily perturbs the coupling between these chlorophylls, okay, these new spectroscopic states are formed, and actually these are new light harvesting states that are formed. So these organisms use a different strategy to ensure robustness to static disorder. Okay, a couple of months ago, a preprint was published on a cryo-EM structure, so resolving the structure of this light harvesting system. So it's only a preprint that is available, very fresh results. And it showed that this chlorophyll, which is a key chlorophyll. So actually this same motif one has in LHC2. So these are 2 homologous systems. And in LHC2, this coupling is extremely strong. This coupling has to be broken in diatoms. And the coupling is broken by diatoms having this chlorophyll, which is chlorophyll A in plants, replace it with chlorophyll C, which breaks this excitonic coupling. Okay, so this actually confirms the model and the results that we have proposed 5-6 years ago. Okay, all these experimental results that I showed you were based on single molecule spectroscopy. There are in fact a whole gallery of different single molecule spectroscopy techniques that are featuring on many different time scales. So on the slowest time scales, in the order of seconds, one can resolve full spectra because one has enough photons in order to resolve the spectra. Going lower, one can resolve different intensity fluctuations. Then one can get microsecond information by modulating the incident excitation light. One can also use correlation principles in order to get lifetime information, so photoluminescence or fluorescence lifetime, and one can also use so some other ways to get information down to the femtosecond time scales. Okay, and this can be combined with different types of photon correlations, polarization dependent. So often one gets multidimensional data, so spectral data, polarization data, intensity data, all of that, and that gives a lot of information about the system as a function of time. Okay, so at the beginning of this year, Nature published a paper where they selected a few tools and techniques that they thought would have significant impact on science in the coming year. And on top of that list, very interestingly, was single molecule protein sequencing. This is a technique that's based on single molecule fluorescence, and there are different variations of nanopores, but some of them making use of plasmonic nanopores. And this technique was on top of the list, even higher than James Webb Telescope, some electron microscopy techniques, CRISPR. So that was very interesting to see that on top of the list. And yes, we are working on single molecule fluorescence techniques, and we have also worked on plasmonic systems. And now I would also show you a combination between these two. So using or integrating LHC2 with gold nanorods in order to control some of the photophysical parameters. Okay, so if the dimensions of a metallic particle is smaller than the wavelength of the incident light, then the incident electric field is going to lead to a coherent oscillation of the conduction electrons on the surface of the metal. Okay, and that will form so-called localized surface plasmon resonances, which have a lot of interesting applications. Okay, so say one focuses light on a gold nanorod like this, and due to the curvature of the rod, so-called lighting rod effect is formed. So you have a large charge concentration here at the tips of the nanorod, and that leads to a large concentration of the electromagnetic light at the tips, forming so-called hotspots near the surface. Now, if you bring a molecule, for example, our LHC2, close to the hotspot, so far away, it's not going to interact with the plasmon field, but as you get it closer, the emission intensity is going to be enhanced significantly because of this exciton-plasmon coupling. Reaching an optimal level, and then when you get very close, in the order of a few nanometers, strong quenching is going to happen because of electron transfer from your fluorescent molecule to the plasmonic system. Okay, and yeah, what makes these systems very interesting is that the spectroscopic properties are highly tunable. So especially if you tune the aspect ratio by changing the longitudinal component, then one can strongly tune the absorption and the fluorescence properties of the system. Okay, so we synthesized gold nanorods of a certain aspect ratio such that the absorption spectrum was in resonance with the absorption and fluorescence of LHC2, which you can see there. And then we used layer by layer assisted methods in order to first put the nanorods on the glass substrate and then spin coating LHC2 in between them. And then we would do single molecule spectroscopy on LHC2. So what you would typically find is that when LHC2 is far away from a hotspot, you would see relatively weak fluorescence. But when it's near or inside a hotspot, you will see very strong fluorescence. And this is illustrated here. So LHC2 in a hotspot, you see a significantly enhanced fluorescence intensity compared to LHC2 that is far from a hotspot. So 2 orders of magnitude increase in the fluorescence intensity. Okay, so this means that the radiative rate is significantly affected by the presence of the plasmon field. But the non-radiative rate is also significantly affected. So here is the enhancement factor, the fluorescence enhancement factor, and the lifetime. The non-radiative rate changes the lifetime. So the higher the non-radiative rate is, the shorter the lifetime is. So here are some LHC2 that are far away from a hotspot, and when they are coupled to the plasmon fields or inside a hotspot, you get a significant enhancement in the fluorescence and a concomitant decrease in the lifetime. So now the plasmonic fluorescence enhancement factor is a product, well, more or less, approximately a product between excitation enhancement and emission enhancement. Okay, and one can decouple these two in order to get for our enhancement, the amount of emission enhancement versus excitation enhancement. So most of the enhancement is excitation enhancement due to the concentration of the light in hotspots near the surface of the metallic nanoparticle. Okay, and one can also get a figure of merit. So if one starts with a very low intrinsic quantum yield, then one can in principle get a very large plasmon fluorescence enhancement in order to get a similar figure of merit when comparing different systems. So plasmonic fluorescence enhancement is especially very useful when one is working with a system that is having a very low intrinsic fluorescence quantum yield. Okay, we also proceeded to model this, and I'm not going to go into a lot of detail, just a single slide on that. So our LHC2, even though it contains 54 pigments, it can still be modeled as a dipole point source. And that is because 90% of the emission comes from the lowest exciton state. And this lowest exciton state can be considered to be a dipole, an emitting dipole. So we consider our whole complex to be a single point dipole antenna. And the rates of relevance are the absorption rate, the non-radiative decay rate, and the radiative decay rate. Now, when this dipole antenna couples with a nano antenna, a plasmonic nano antenna, then one gets excitation enhancement. So one will have to multiply this absorption rate with an enhancement factor. The radiative rate is also enhanced by the so-called Purcell factor. And there's an additional non-radiative decay term that arises from energy transfer from our molecule, like LHC2 in this case, to our metallic nanoparticle. And most of this energy that's transferred is then thermally dissipated in the metallic nanoparticle, causing Joule heating. So that is also non-radiative. Okay, so from this one can calculate the fluorescence quantum yield, and one can model the absorption and fluorescence spectra. So we've done all of that. Not going to give you all the details in this specific talk. Just the last two slides before I'm done. So single molecule spectroscopy can reveal a lot of detail, very interesting information, but one must always take into account that it is a pretty unnatural environment. Firstly, one has to isolate these complexes, these pigment protein complexes from their natural environment, and then dilute them significantly, study just one at a time, and they must be attached or they must be immobilized in some way in order to collect enough photons in order to resolve spectra or other dynamics. So typically they are attached to a surface, like in this case, or they are confined by a plastic by spin coating them in PVA, for example. Okay, and these methods can all perturb the structure. And if you remember the example I showed you a few slides ago of the difference between PS1 and PS2 complexes, a perturbation that causes a change in the distance between two chlorophyll rings of less than 1 angstrom can have a drastic effect on the function. So this is the main reason why we are developing methods to go closer towards in vivo single molecule spectroscopy and to avoid confinement by plastic or attachment to a surface that can perturb the protein structure. And for this reason, we have developed, for example, real-time single particle tracking. And the purpose of this is that one can follow the diffusive dynamics of a particle or a complex in this case in real time, and on top of that do spectroscopy or imaging, anything like that. So in this way one does not only avoid the immobilization aspect, but one can also get diffusive information which can give you size information and one can get superior spatial temporal resolution, give you an extended tracking range to follow, three-dimensional tracking and all of that. So that is one of the directions that we are strongly working towards at the moment. And we already have provisional experimental results. Here's a theoretical study on this method and also a review paper that we published. Okay, so this concludes basically what I wanted to say today. I wanted to display these few take-home messages, and if you can identify other take-home messages, then I'll be very glad. So let me quickly read these few bullets for you. Firstly, quantum biology is, in my opinion, a very exciting and emerging research field and an interesting and promising playground for the physicist. Secondly, single molecule spectroscopy is a very sensitive and selective method for studying the rich dynamics of proteins. Photosynthetic proteins sample their disorder to establish their biological function. And I've showed this specifically when comparing the spectroscopic difference between photosystem 1 and photosystem 2 systems. And then I highlighted a lot exciton states, charge transfer states, and exciton charge transfer mixed states. And all these types of states are used by light harvesting complexes in ingenious ways to improve their light harvesting and photoprotective functions. So in this talk, I have not talked about the photoprotective functions, but they also play an important role in various aspects. Okay, and then lastly, metallic nanoparticles are very useful for investigating especially weakly fluorescent molecules. Okay, so I would like to acknowledge especially the team of students, postgraduate students that are behind all the results that I've showed you. This is actually an old group picture at our single molecule spectroscopy setup, a home-built setup, just across my office here at the University of Pretoria. So besides them, all the new students, all collaborators that were involved, all the funders, without which none of this would be possible. And thank you very much for your attention, and I would be very glad to answer any questions.
Yeah, so thank you, Professor Kruger. It's a really wonderful talk, and it's a crystal clear presentation that you have given. So I felt like that I'm in a classroom learning a lot of things, okay? And the interface of physics and biology, a lot of, in-depth understanding. So now I request all audience to interact with, Professor, with your questions, queries. Maybe I will start with one. So if maybe Professor Deshmukh, is this fine?
Yeah,
I have
please.
two queries actually.
Please go ahead. Please go.
So Professor Kruger, so the first part of your talk, you talked about particularly the chlorophyll, the splitting. The low energy band, which is around 800 nanometer. Typically in the textbook, we learned that is a QS, QX, QY kind of nomenclature people give, at least in chemistry audience. And you mentioned about that splitting is basically due to something polaron and exciton coupling. Can you just, little bit elaborate on that? That about 50 nanometer splitting, if I remember.
Yes. Okay, so yeah, thank you very much for the question. You have touched on different aspects. So these spectra are much more complex than the picture that I've given. So one is considering these tetrapyroles, and actually in chlorophylls you have a broken symmetry because one of the double bonds has been reduced. So whereas one has normally two equal molecular axes, in this case the two molecular axes are now different. So that leads to a splitting between the QX and the QY absorptions, the transition dipole movements. Okay, but this is different from the exciton splitting. Okay, so one gets this splitting between the QX and QY in any case. Exciton splitting is different. That gives rise to an additional splitting. And that is due to the coupling between different molecules that is causing this excitonic state, delocalized excitation state, that leads to excitonic splitting, so splitting of the energy levels. And what I said in that one example, so that was LH2 of purple bacteria, the energy difference between the lowest and highest exciton states, there I'm talking about exciton splitting, that is 50 nanometers. So just to illustrate that the exciton splitting can also give a large splitting and a large energy gradient.
I see. Thank you. Now it is clear actually, crystal clear. I was confused with the QY splitting, and that is due to the symmetry, symmetry broken systems. I see. And the second question, Professor Kruger, that I have. So at the middle of your talk, you talked about the chlorophyll fluorescence quenching has been explained in the literature, probably based on the presence of the charge transfer excited states or the coupling of the charge transfer states with the molecular excitons. So I'm just wondering, what is the origin of this charge transport states in this kind of pigments like chlorophyll? Who is, is acting as a electron donor, which is acting as an electron acceptor? Is it intramolecular or intermolecular? I'm a bit, not clear. It's not a typical donor acceptor type of molecules, but still you talk about the presence of CT states in order to explain the fluorescence quenching, right? So can you just,
Yes, thank you. That is again a very good question, and also touching a lot of aspects. So the example that I used of solubilized free chlorophyll, it means that we only have chlorophyll, well, chlorophyll A specifically. So they are supposed to all have the same energy. But now, because of the very high density, in the case of concentration quenching, these molecules will all move around, but when they collide, they will transiently overlap their wave functions, and that can give rise to the formation of charge transfer states. And because of the very high density, these transient states form very quickly. Very frequently. Okay, so charge transfer states are they are always present. It's one possible state, but their energy is not always such that they can be populated. So what then happens in these systems, I think during the collision process, or in any case for chlorophylls, the energy of the charge transfer states are not too far from the S1 energy level. Okay, so when some distortion occurs to bring the energy levels closer together, they can mix, or they can be an energy transfer from the S1 state to the charge transfer state. Charge transfer state is a dark state. So once that is populated, you will get thermal dissipation. Okay. In the case of concentration quenching, there's no mixture between the electronic state and the charge transfer state. So it's just the energy transfer to the charge transfer state, and that is then that leads to complete quenching of the energy. In the examples that I showed of the light harvesting complex, in that case, there was a mixing between the exciton and the charge transfer states. So because of this mixing, you still have oscillator strength from the excitonic state, and that leads to fluorescence. Okay. And you get some quenching because of the involvement of charge transfer state. So to say technically correctly, if you remember the one equation that I showed, maybe I can just quickly get back to that.
I think it's like a AV coefficient, the state 1, state 2 mixing that you talk.
So there are actually two hybrid states that form. Let me see, where are we now? This one. Okay, so you get an exciton state with some charge transfer characters. So it's mainly an excitonic state, and it's mixing with a little bit of charge transfer state. So this will be the state with a high oscillator strength. So that will be the emissive state, and normally this is the state that is low enough in energy that will be populated. This other hybrid state is normally much higher in energy than the exciton state, so that will not be populated, and that is a charge transfer state with some excitonic character, but because it's not populated, it is actually not relevant. Okay, so because of the presence of the charge transfer state, charge transverse state has a strong permanent dipole moment. Okay, and that leads to a very strong Stokes shift because of the large reorganization energy. So one has large oscillator strength and a large redshift. And this is exactly what we also see in the experiments.
Nice. Very good. Thank you so much, for explaining. So others, audience.
I have a couple of comments.
Yeah. Professor, go ahead. Yeah.
Tjaart, it is such a fabulous talk. First of all, thank you very much. Tjaart, I have a few, one or two confusions in some of the points, but I would like to clarify. When you when you talked about the metal nanoparticles, you get the localized surface plus bond resonance. Okay?
Yes.
As you mentioned, of course. And in one of the graphs, I have seen that you have made a plot of the localized surface plus bond yield as a function of the wavelength. Okay. And I've seen each one has the same intensity at various wavelengths. Does it make sense? You are getting all the same intensities of the localized plus bond resonance field at all wavelengths. What is the physical, significance of that?
Which slide are you exactly referring to?
Well,
49.
I think you have shown some LSPM as a function of wavelength.
See this one. So I think this is the only slide.
Yeah,
as far as
probably you can repeat that also. Why the intensity is same exactly at different.
Okay, so there is a strong wavelength dependence. We've actually showed it in our modeling results.
Exactly.
And that is mainly because of the resonance between the absorption of the gold nanorod and the absorption and fluorescence of LHC2. Because one should consider this to be like a donor-acceptor system, energy transfer from LHC2 to the metal nanoparticle, but also other way around. It's a two-way antenna system. A double antenna system, and it is strongly wavelength dependent. It strongly depends on the resonance between these different bands. So yeah, it's shape dependent. This is actually one of the reasons why we saw a heterogeneity in the results that we found, because of different shapes and sizes. That is going to change the spectroscopic properties. So now, because of this resonance, there's both an absorption and excitation and emission enhancement. Okay, and especially the excitation enhancement, that is going to depend significantly on the resonance between the absorption properties of the metallic nanorod and the absorption and fluorescence properties of the molecule, the complex that we are studying. Okay, so there must be a strong wavelength dependence. Okay. I have not included our calculated results there. We have modeled all of this, but yeah, I can send that to you separately if you are interested.
Other results, we always had this, the distillate the fields at different frequencies.
Yeah, sorry, Professor, I could not hear you just now.
Yeah. Oh, can you hear me now?
Yeah, Purushottam, you need to get closer to the microphone.
Yeah, okay. Oh, can you hear me now? Can you hear me now? Can you hear me?
Yeah. Please go ahead. Your microphone is
Okay,
on side. Yeah. Now it's fine. Go ahead.
I can ask you one more question. When you talked about the charts of the
No, Professor Purushottam, again, probably
yeah, your voice is breaking.
Questions. So
your voice is breaking, Purushottam.
Oh, I don't know why it is breaking. Can you hear me? Loudly.
Or maybe you can put your question in the chat box.
Okay, all right. Oh, anyway, I can even talk later. No problem.
No, whenever your face is on the left side or right
Yeah.
side, I see you are we are getting okay.
Yeah, well,
Yeah, you can
well,
speak like that. Yeah.
well, I'm not moving that way. Anyway, but anyway, now can you hear me? Can you hear me?
Yes.
Can you hear me now? I
Yeah,
don't
so
know.
No.
Professor, yeah, at the
Yeah, I
beginning, I
think
can always hear you, but when your face moves to the other side of the screen, can't
Oh.
hear you anymore. Yeah, so if you
absolutely,
stay in
yes.
that position, then you're audible.
Oh,
Yeah.
Yeah.
So
Yes.
I didn't understand one point. This is the olfaction. Could you explain what was olfaction?
No, Professor, again, you are, moving your face far. I mean, far from your microphone, probably.
Okay, then I think I keep my face down. Is it okay?
Maybe you can type. I can, read out and
Okay,
Yeah.
yeah.
okay. It's
Anyway.
okay. It's okay. It's not very clear.
No,
It's
no.
okay.
When you say it's okay, now we are able to hear you. I mean,
No, I was asking that. Can't you hear? Okay. Olfaction. Can you hear that?
No, I'm afraid, Purushottam, we are losing some of the
I
words.
don't know. I don't know what is happening. I don't know what is happening. I don't
So we are not able to follow the
Okay.
conversation.
Okay. I can.
I think either you type it in the chat box or maybe correspond later by email.
Yeah,
I
yeah.
think.
I will do that. I will do that.
Okay.
Anyway, thank you very much, sir.
Yes. Arun, I think if there are no further questions, I don't know if there is anything on the YouTube or this thing.
Can I?
I'm checking. I have checked. Actually, there is.
If you can hear me, I can ask another question. Can you hear me?
Yeah, now you are audible clearly.
Yeah, absolutely.
Okay, Zart, I was asking about how could you detect the single photon? Are you using the photomultiplier tube? I mean, just a very basic question.
Yes, so one could in principle use a photomultiplier tube, but the quantum efficiency is very low. So we are using single photon avalanche diodes, so single photon sensitive APDs, and they have typical quantum efficiencies of 70% or more.
I see. Under another point, I'm asking under what condition you have the charge transfer between the excitons? Is there any specific condition for that?
Charge transfer. Okay.
Yeah, you talked about charge transfer between excitons, because exciton is a quasi-particle, which is a neutral particle, But we talked about the charge transfer.
Yes. Yeah, that actually relates to the question of Prof. Mana earlier, that these are identical molecules in chlorophyll A. So if one considers, for example, the reaction center, so the first step of photochemical charge separation is electron transfer from one chlorophyll A to a neighboring chlorophyll A molecule. They are located, oriented such that their rings are almost parallel, about 1 nanometer apart. Okay, so that gives perfect conditions for charge transfer. And then the local protein environment would change the redox potentials to make one of these two chlorophylls specifically a donor and the other one an acceptor. So this is one of the key roles of the protein environment. That is why I said at the beginning of the talk, the protein is much more than a scaffold. The protein changes the spectroscopic properties of each chlorophyll, of each pigment. So in fact, when you would put a chlorophyll at different positions inside the protein, the spectroscopic properties can be drastically different. It's because the localized dielectric constant is different. Protein is a highly charged substrate. Okay, so yeah, this is also what happens at the first stage of charge separation. In the case of the concentration quenching in solution, I have not investigated that in detail, but I suppose due to the collisional effect, there would also be a temporary change in the spectroscopic properties of the two chlorophylls, and then one of the two interacting chlorophylls would act like as the donor and the other one as the acceptor. And this would then be a transient effect.
Okay, thank you. Thank you.
I think, Professor, there are no more questions. So maybe, yeah.
Well, thank you very much, Arun and Purushottam, for conducting the session, and great privilege for us to have Professor Kruger for today's talk. You covered so many intricate aspects of biophysics and so many interdisciplinary important areas of quantum biology. It's amazing. So I am sure that your talk will be watched on the YouTube. And I should mention that the center, the CAMOST, which is hosting this seminar series, is jointly hosted by two institutions in Tirupati. One is IIT, which is dedicated a little more toward engineering, and ISER, which is a little more tilted toward basic sciences. And I have had the opportunity to interact with students from both the institutions. And I have always been saying to the ISER students that they're very privileged to have courses in their first 2 years. You know, when they are fresh out of high school, they're exposed to basic sciences as well as mathematics and physics and chemistry. And I feel that getting that exposure when they are like 18, 19, 20 years old is a great, great privilege. And like you said, this is the century of, you know, biophysics, and, you know, you definitely need strong background in physics, chemistry, and biology and life sciences to contribute toward advances. And these, it was amazing that you pointed out that this is the most important area which has been identified by several academicians. So I think it is very impressive, and that throws up great opportunities. And I really think that your talk has been absolutely scintillating, and I'm sure it will attract many young students to this field. So thank you very much. And thank you, Arun and Purushottam, also for wonderfully conducting the session today. Thank you.
Yeah, thank you very much. Yeah, it was a really nice, wonderful to have Professor Kruger, and very nice. Yeah.
Yeah, from my side also, thank you very much again for inviting me. It was a pleasure to interact with you. And please feel free to email me after the talk. And that also applies to anyone watching this video. Please feel free to email me. I don't think I have given my email address, but if you just Google University of Pretoria and add my name, you'll be able to find my email address very easily.
Yeah, we should be able to find it.
Please feel free to ask me questions over email. I would be very glad to answer them.
Okay, so thank you all very much, and I would say goodbye for now.
Yeah, goodbye to all.
Very much.
Bye-bye.
Yes,
thank you, Tjaart.
Thank you very much.
Yeah,
Thank you once again, Tjaart. Nice to see you.
Yes, nice to see you too.
Yes, goodbye.